Ladies and gentlemen, good morning. I'm Dr. William Zhang. I'm from Vector Builder. I'm an application scientist and I am in charge of the CRO department, which is developing this powerful directed evolution platform for novel AAV vector discovery. Today, I'm very glad to join this seminar, and in the coming session, I will give you a brief introduction to our powerful platform, AAV capsid evolution. So let's review some basics about AAVs. They are very small, non-envelope viruses, and their single-stranded DNA genome is encased by the protective protein shell called capsids. And thanks to AAV's broad tropism, prolonged gene expression, and non-pathogenicity, and low immunogenicity, recombinant AAV is a highly popular gene delivery vector for a wide range of gene therapy and vaccine application. As its application is becoming more and more mature, however, several problems with existing AAV serotypes limit their therapeutic potential, such as uncovered and undesirable tissue tropisms, as well as low yield, low transduction efficiencies, and pre-existing neutralizing antibodies, etc. As we know, AAV make their first contact with the target cells through their capsids, and that's one of the most important factors that makes AAV so special. Over the past decades, scientists have led to an intensive focus on optimizing AAV by capsid engineering. So to overcome existing AAV limitations, capsid engineering by directed evolution has been a critical area of research that has significantly accelerated the development of new AAV variants with improved features. This process typically begins with the generation of a diverse library of AAV capsid variants. This library is then subjected to selective pressure, such as exposure to target cells or in vivo environments, to identify variants with improved transduction efficiency, altered tropism, or enhanced resistance to immune response. The selected capsid variants serve as the basis for the next round of evolution, and this iterative cycles continues until a variant with optimized characteristics is obtained. Directed evolution has proven instrumental in tailoring AAV vectors for various gene therapy applications, allowing researchers to fine-tune the capsid features to meet specific therapeutic goals while minimizing undesirable side effects. We support library construction by diverse approaches, such as screening in multiple species, including mice and NHPs, followed by in-depth data analysis. To elaborate, I'm going to introduce several main approaches to generate an AAV capsid library, namely the random peptide display, error-prone PCR, and DNA family shuffling and in silico design. Before that, I'd like to briefly introduce the AAV packaging system that we use in this AAV capsid library packaging. So it's very similar to traditional non-capsid library AAV packaging. We also use the triple or double plasmid transfection system to do AAV packaging in AAV capsid evolution projects. So only one difference is the transfer plasmid. In this kind of capsid evolution project, we use library plasmids instead of a wild-type serotype of AAV capsids. So we would also include the helper plasmid and RAB expression plasmid to do triple transfection in HEK293 T cells to do virus packaging. So let me give you an example for random peptide display. So let's take AAV9 for example. In this method, random peptides of various lengths are inserted into a variable region of AAV9 In this slide, I am showing the Q588 amino acids is the position for 7-mer random peptide insertion. This technique could potentially allow for generation of AAV serotypes with different tissue tropisms, as the randomly displayed peptides may alter the ability of the AAV capsid to interact with the specific cell surface receptors. By this method, researchers can potentially capture capsid mutants capable of modulating AAV tropism and transduction efficiency, and eventually optimizing gene delivery to the specific tissue or cell types. For error-prone PCR method, we generate a capsid library by adjusting the PCR conditions and parameters so that we can introduce random mutations in the full length of a particular capsid gene, resulting in a complex library. However, this approach has shortcomings when it comes to knowing the full length of the mutants due to the NGS limitation. As we know, the NGS allow us to sequence 150 to 250 in length of the target gene region. If the mutations happens at the full length of the capsid gene, which is about 2K, the NGS has its limitation to know the full length sequence of a specific mutant. So we have to balance the choice based on the goal of your research. So the next one is DNA shuffling method. This method, the capsid genes of various AAV serotypes are digested, followed by re-annealing through homology, resulting in the generation of new serotypes that are hybrids of their parental capsids. These resulting capsid mutants offer a diverse repertoire of AAV variants, and each possessing unique combinations of genetic traits derived from multiple serotypes. So as we know, such as AAV 2.7M8, which was reported in a research several years ago, we have observed this method is very powerful to generate a novel capsids that facilitates the photoreceptor transduction in vivo. Then let's come to the in silico design. So for this method, it stands out as another powerful technique that employed for the generations of the capsid libraries. Unlike experimental methods like the previous one, such as DNA family shuffling, in silico designs operates in the realm of computational biology, leveraging properties, proprietary algorithm and simulation to predict the engineer the AAV capsid variants with desired properties. In recent years, machine learning has become a very popular and identifying new serotypes, showcasing the effectiveness and systematic mutagenesis and machine-guided protein engineering. So these methods have their pros and cons and in different contexts and applications. So Vector Builder can help you tailor the experimental design and choose the most suitable strategy to work with. So let's look at a case study to see how Vector Builder's AAV capsid evolution platform can help you to generate a better blood-brain barrier crossing novel capsids. So in this study, we aim to find novel capsids that have enhanced BBB or blood-brain barrier penetration properties. And we utilize the random peptide display method to generate the AAV capsid library. So in the first step, we generate the plasmid library, and then we do NGS sequencing to validate this library and to QC this library. And from the data I'm showing here, we see the plasmid library is relatively even for each variant. And after virus packaging by our proprietary optimized methods, we have found some bias in the variants that results from the packaging preference for different mutants, because we expect different mutants have different capabilities of virus packaging. So we would expect to see some bias have begun to show during virus packaging. Well, after we subject the virus library to NGS. And then after that, we use the packaged virus to do in vivo screening. And then we will harvest the target tissues, such as CNS tissues in this study. And we found very biased mutants have been enriched or depleted in different tissue types. So these are the basic of this kind of study. And also, we analyzed the stop codons during the virus packaging, particularly for the first library, because for a 7-mer random peptide display library, we would introduce NNK codons, and these codons contain stop codons. And stop codons theoretically could not generate a viable capsid protein. But in fact, we detected the stop codons in the virus library. That could be resulted from sequencing error or cross-packaging. And this is a very important or critical parameters that would affect the quality of this virus library. So we look into this parameter of stop codon percentages in virus library, and then after evaluation, we would find out whether this batch of virus would be suitable for downstream assays. So after NGS, we would have the different peptide or coding sequence data from the NGS data, and we will do analysis during comparison of the amino acid frequencies in the plasmid and virus library. We would find different amino acids would appear in different occurrence or frequencies. So to desensitize the data, the amino acids shown here are masked by numbers. We categorize them by their nature. Detailed analysis of the amino acid species and positions give us a more in-depth understanding of sequence bias at the peptide level. The red triangles indicates the baseline of enrichment, and the bars show enrichment degrees. So as shown in this diagram, favored and disfavored amino acids have different enrichment profiles. By enrichment analysis after virus packaging, we can have a primary picture of the direction of sequence evolution regarding the pro-packaging capsid variants. So similarly, after screening in vivo, peptide sequence that favor target tissue transduction enrich more, and they also showed a more biased enrichment at specific positions. These data give us a better chance to identify the tissue-specific motifs, which are significant and for candidate selection used for the next round of screening and validation. So we also compare the overall amino acids preference at each site or each library, such as the plasmid, virus, and tissues. Let's take a certain amino acid, for example. We observed a different enrichment in different libraries, and then we speculate that due to the different nature or properties of a certain amino acids, it will behave differently in the virus and tissue library, because some of them might be pro-packaging, but not pro-transduction. But if some of the amino acids enrich in the tissue library, that it might help the transduction enhancement very much. So yes, we will have a better understanding of the amino acid frequencies in different libraries, and to facilitate us to narrow down a second list or third list for candidate selections. So after in vivo screening, we will harvest the tissues from the animals. During this research, we do systemic injection of the virus library by tail vein injection. As we know, these AAV particles have to cross the blood-brain barriers to get to the CNS tissues, such as the brain tissues and the spinal cord tissues. If they are not BBB penetration, they would be selected, or they will be, during the evolution, they will be depleted. And then after harvest of the tissues and NGS, we will have the candidates in different tissue libraries, and then we do a Venn diagram to overlap some of the candidates, and then we can select some of the candidates that are suitable to go for the second round of screening. We also rank or do scoring for the peptides or the mutants, because after screening in vivo, we would expect different peptides would behave differently, and then we will have a better understanding of which peptide or which mutant would be more pro-transduction in the CNS tissues, which indicates it has better capability of BBB penetration. And after this enrichment analysis, we'll compare the mutants with the wild type. Let's normalize the wild type as 1, and we will get the score of different mutants. And the higher the score, the better the mutants. And the next round would be the validation in vivo. So after mutant selection, we will individually package the AAVs with these mutants. We change the transfer vector with reporter expression cassettes. We will package these virus with the capsid mutants in the packaging system. We use constructed capsid expression vector to do packaging, and after that we can validate different mutants individually. After virus packaging, we will inject the mice with these mutant capsid AAVs, and after that we can find different sections of the mice brain tissues would have different reporter expression patterns. For this case study particularly, we found that CAT4 mutant would generate a better EGFP expression signals, especially compared to the AAV9 wild type, because we derived this mutant from AAV9, and this mutant now has demonstrated a better BBB penetration capability to let the AAV transduce the brain tissues successfully and give us a better observation of the EGP signals. And moreover, we also do the validation in the spinal cord tissues. We harvest spinal cord tissues and do sections, and then we check the EGFP expression level. Compared to the AAV9, we found these four mutants, CAP1, 2, 3, 4, are all capable of penetrating the BBB and then transduce the spinal cord tissues. But as we know, these are the I'm sorry. I'm showing the liver tissues. So these are the spinal cord tissues. And let's get back to the liver tissue. As we know, the AAV9 is capable of transducing the liver tissues as well. But in this study in particular, we actually don't want the mutants to be that powerful of transducing the liver tissues, because these are considered as off targets. So let's look at the CAP2. We see the CAP2 mutants have a less capability of transducing the liver tissues, which is also good for de-targeting the unwanted target tissues. Because during application of an AAV vector, we always want the AAV vector to target our target tissue very successfully and to target our unwanted tissues at a very low efficiency. So this would be a balance to choose between the target tissues and non-target tissues. So here are the spinal cord data. We also found that CAP4 and CAP2 are very good at transducing the spinal cord tissues. So these are the lumbar spine, and this is the cervical spine. We look at different sections of the spinal cord. So compared to a wild-type AAV9, we can show you through our platform of AAV evolution, we can generate novel mutants that can be more capable of BBB penetrations compared to wild-type AAV9 capsids. So I'm showing you the next case study to discover a novel AAV capsid for enhanced photoreceptor transduction via the intravitreal injection. It's also done in mice. The administration route will be different. It's not from the tail vein injection. We did intravitreal injection, which is through the tissue of the eye. And we sampled the retinal tissues 1 month post-injection. So we compare these mutants with a established AAV 2.7M8. Through a similar method, we generated the library, and then we do virus packaging, and then we do in vivo study. And when we do virus packaging to validate the mutant's capability, we use this transfer vector for reporter expression. We constructed an CAG NLS mCherry transfer vector to let us know that when the AAV is successfully entered the target tissues, we can observe the mCherry signals in the cell nucleus of the target tissues. So this A2C38 is our novel capsid, and this AAV 2.7M8 is the published or reported the mutants constructed from AAV2. So yeah, here I'm also showing you at a much lower dose for our mutants, we can achieve a similar transduction efficiency. So this is a AAV 2.7M8 result in vivo, and this is A2C38 result. As when we compare these two mutants, we see very similar transduction efficiency in vivo. So, but for our mutants, we injected a much lower dose. So this would greatly enhance the safety of the application of these capsids in the future if therapeutic goals are meant to be applied for these capsids mutants. And then this case also shows us show that our platform is very powerful to generate novel mutants that are, you know, at least parallel to existing mutants, or even better in terms of safety during the novel capsid discovery. And that's all my sharing for today, and I'm very happy to take questions. Thank you.